Hello everyone, this is our team talk on storing information on the paper Brief Novel Visual Experience Fundamentally Changes Synaptic Plasticity in the Mouse Visual Cortex. So in this experiment, the researchers were studying mice, and it had been previously established that there was a long-term potentiation and long-term depression dependent on an NMDA activation. NMDAR is a glutamate receptor, and it was understood that dark exposure, so mice raised in the dark from birth and dark rearing enhanced this NMDAR dependent long-term potentiation. So the question was that what changes in plasticity will be evoked by exposing light to mice that had been reared in the dark since birth. So methods, there were three groups of mice. First, they were normally reared, which were reared on 12 hours of light, 12 hours of dark normal schedule. Dark reared, which are only exposed to dark their entire life. And then dark reared and then light exposed. And dark reared were same as dark reared mice, the previous group, but then they were exposed to light for different amounts of times prior to them being used for the experiment. And so they used visual cortical slides, so containing the V1, the visual cortex, and specifically the layer 2/3 pyramidal cells. And so to test this long-term potentiation, long-term depression, they evoked postsynaptic current recordings using a voltage clamp method. The conditioning stimulus for all of the experiments to follow was 360 pulses at 2 Hz paired with continuous postsynaptic depolarization, and they used a variety of drug solutions, both receptor blockers and enzyme antagonists, to control each experiment. So figure 1 is titled Novel Visual Experience, which unmasks the NMDR independent LTP. So the researchers did their conditioning stimulus on all three groups of mice. So we have the dark reared, normally reared, and light exposed mice. In A, we have the normally reared mice, and that pairing induced a robust long-term potentiation, as evidenced by the dark filled data points, and that was in artificial cerebrospinal fluid, so no real drugs are inhibiting it. And then in the open data points is APV, which is a glutamate inhibitor, and we see that the NMDAR receptor is inhibited and there's no long-term potentiation. In graph B, we have the dark reared mice, and they also show a robust long-term potentiation, even stronger than in the normally reared mice. And they also have the same lack of long-term potentiation in the APV, which suggests that there's a similar mechanism which is occurring in both the dark reared and normally reared mice. And in graph C, we have the dark reared and light exposed mice, and interestingly, these mice exhibited a long-term depression under normal circumstances and a long-term potentiation in the APV. So this suggests that the visual experience after dark rearing, but in that light exposure, has a profound change in the synaptic plasticity of those neurons. And also it reveals a new NMDAR independent form of LTP, because we know that the NMDAR receptors are inactivated in APV. So when we see this long-term potentiation in C, we know that there's a new type of mechanism that is responsible for that. And finally, in D, this is a statistical graph that shows that the pairings were robust and of significance in the majority of the mice. So in figure number 2, the researchers explained that how light exposure restores some normal NMDA receptor subunit composition at the synapse. So the focus of the experiment is on the NMDA receptor independent form of LTP in light exposure. However, the researchers want to check if alterations in the NMDA receptor dependent form of LTP and LDT is due to changes in the subunit composition after light exposure. So it's been previously established that NR2A and NR2B subunits at the synapse change with experience-dependent manner. So the researchers measured the fraction of NR2B subunit at the synapse in all three conditions: normally reared in white, the dark reared in the filled data points, and the light exposed in the gray data points. And they also introduced ifenprodil, which is an NR2B antagonist, and then they measured the NMDA receptor response and the change in the decay constant. So in the dark reared fraction In dark reared, the fraction of response blocked by IF and Prodel and the decay response was larger, which correlates to a large proportion of NR2B subunits. In contrast, the light exposed and normally reared mice had similar levels of NR2B, and this suggests that a rapid restoration of the normal protein subunit proportions in the light exposed population. So it took very little amount of light in order for the dark reared mice that were light exposed to obtain the correct levels of subunit composition that we see in the normally reared mice. So this therefore argues against the role of an altered NMDAR receptor function in the changes observed in long-term plasticity and long-term potentiation and long-term depression after light exposure, because we saw that the subunits restored at similar levels.
In this part of the experiment, layer 4 cells were stimulated to create EPSCs on layer 2-3 cells in dark-reared mice with 10 to 12 hours of light exposure. These experiments were done in APV, which is the NMDAR antagonist, and they used the whole cell voltage clamping technique. In the graphs, we have EPSC amplitude in percent of original amplitude over the time in minutes. In part A, the APV blocks the NMDAR response to show the novel form of LTP, which is a depolarization response, and that's shown in the closed circles. mGluR5 antagonist MPEP blocks that response in the open circles, so that the LTP is not observed. In part B, there's no blockage of the novel LTP when other types of mGluR antagonists were used, such as mGluR1 antagonists. In part C, DHPG, which is an mGluR agonist, causes the persistent LTP to be shown in these closed circles, which can once again be blocked by the MPEP, mGluR5 antagonist, which is shown in the triangles. In part D, the calcium chelator BAPTA sequesters calcium in the postsynaptic cells, or the layer 2-3 cells, and blocks the novel LTP, which is shown in the open circles here. Overall, we see that mGluR5 seems to account for the novel LTP as opposed to other types of glutamate receptors, and that the response is dependent on postsynaptic calcium increases. In Figure 4, they use the same process, but with more blocked enzymes that they hypothesize might be mechanistically important to the induction of LTP, rather than the receptor properties of this response. mGluR is a metabotropic glutamate receptor, and so we might expect the mechanistic steps to include protein kinase C, CAMK2, or calmodulin-dependent protein kinases, and protein synthesis through the translational enzymes. They use the same procedure in dark-reared mice with 10 to 12 hours of light exposure, and they did the experiments in APV, which is the NMDAR antagonist again. In parts A, B, and D, they inhibited protein synthesis, PKC, and CAMK2 respectively, and they did this using the pharmacological agents. And they saw that the LTP, which is shown in the filled circles, was blocked, and that's shown in the open circles. In part C, they inhibited PKA, which is protein kinase A, and they didn't see a blockage of the response, which means that it's not an important part of the LTP mechanism of induction with mGluR5. Overall, we see that mGluR long-term potentiation that's promoted by vision after dark rearing is mechanistically dependent on PKC and CAMK2 activation, as well as protein synthesis pathways.
So in Figure 5, this graph displays the transient and delayed properties of the metabotropic glutamate R5 dependent long-term potentiation. So there must be between 6 and 16 hours of light exposure in order for a metabotropic glutamate R5 dependency to be exhibited, and this is evidenced by the dark filled circles. So we see that there is a spike between that time, and interestingly, also during that time, there is a transient loss of the NMDA receptor LTP, as evidenced by the light circles. And so this suggests that the visual cortex could support both types of mechanisms, but probably not simultaneously. And there could be some sort of push-pull plasticity between these two mechanisms that results in this independence of each.
In this part of the experiment, they were looking at whether or not the dark rearing is required as opposed to just dark exposure following normal rearing. They found that prior visual experience, in other words being raised in light, blocks the mGluR5 long-term potentiation in both juvenile and adult mice. In A and B, which are juvenile and adult mice, the mice are exposed to dark, and those ones exhibited a larger LTP, shown in the closed circles, than normal reared control mice, shown in the open circles. In figure C and D, the dark exposed mice were re-exposed to light. Unlike the dark reared mice, the re-exposure to light didn't promote the long-term depression. Rather, after the re-exposure, the NMDAR response, which is shown in the closed circles, was simply blocked by APV, which is shown in the open circles. If the mouse is reared in light-exposed conditions, then it doesn't display the mGluR5 LTP. Dark exposure only enhances NMDAR LTP. In other words, dark rearing, as opposed to dark exposure, is required for mGluR5 long-term potentiation. Overall, what the authors of this paper found is that there is a novel NMDAR independent form of long-term potentiation in the mouse visual cortex in layer 2-3 pyramidal cells. This form of long-term potentiation is dependent on mGluR5, which is a metabotropic glutamate receptor. The process of induction of this form of LTP mechanistically involves intracellular calcium rise, PKC, and CA-MK2, as well as protein synthesis. Expression of mGluR-dependent long-term potentiation is transient. It only appears when the mice have been dark-reared from birth and have been exposed to a novel visual experience. The temporary increase of mGluR LTP coincides with the temporary reduction of NMDAR LTP. Together, that evidence suggests that these synapses support both mechanisms of LTP, but not at the same time. There seems to be a push-pull type of co-regulation of these two plasticity mechanisms. It's unclear what the functional consequences of brief increase in mGluR LTP are, but the authors hypothesize that sudden exposure to novel light might recruit dormant forms of plasticity that could allow for a quicker establishment of receptive field properties upon this novel experience of light.